Good evening, everybody. And it's delighted to see all of my teachers, Dr. Venkat, Dr. Swami, Dr. Vyas, Dr. Shairam, and all of my colleagues. And it started with Selva. He just asked some questions and we put some video. And we gathered today. Actually, Dr. Swami, the doctor, is there. We want to learn more in deeper how to solve this problem. What is happening, what Dr. Venkat described and all those things. We are facing this problem almost 2019 December, and we thought of it is coming down, but now it is more than 4 lakhs cases and so many people are dying and all. So after putting this thing, 1 minute. Can you see I got some questions from different things, so I want to put into for all those things to discuss whatever the 40-45 minutes and take the collective inputs and wisdom from everybody. Have to do that. First, questions from where the SARS CoV-2 has been originated. Second, why it's so infectious? How does it infect the body? What are the symptoms? And does it differ from wave 1 to wave 2? And now this buzzword is the single, double, and triple mutant of coronavirus. Why the infectivity rate of double mutants is very high as of today compared to first one. And why the need of oxygen has been enhanced manifold. How to protect ourselves from the COVID infection. The necessity of the wearing mask and how don't allow this virus to infect us. Now the Lancet report hinted something. Is it airborne? Any proof? Not confirmation yet. And now the questions of vaccination, duration, a lot of things are there. And last things what we're going to discuss about, because of people are staying at home, students are staying at home, life has been disturbed. There are so much of indirect problem coming, which is creating stress, and then people are getting anxiety, stress, and the depressed. There are so many other things, how collectively we can come out from that, what could be the solution. And we'll get all of the leaders here today in the meeting to learn more, and collectively we'll do something so that at least we can protect ourselves and save some life for the others. So now, if you see this, I'm just telling this SARS, but this coronavirus is not new. It's an old one. And there will be around more than 5,000 different coronavirus, it's known. And 500 has been identified in vaccine China. If you remember in 2003, the SARS CoV-1 version 1 came. And here I've just given all those statistics. SARS CoV-1, almost 8,098 people was infected, but this fatality rate is very high. But the infectivity rate was not that great. It came from that bat, then back to some other animal, and it goes to the human and goes back. And then in 2015 or 14, Middle East coronavirus came, infected camels to human, and that infectivity rate was not that high, but fatality rate was very high. So why that infectivity was not? And though people tried to develop some vaccine, but the problem was sample size was not there, and the vaccine was in some cases, it's 1 year, 6 months for SARS-CoV-1, but then everybody forgot all those things and all. So this is the things that we know about SARS-CoV-1 and MERS-CoV-1. And now, how it happens, just simply to tell you, from that bat to there is a pangolin, and then pangolin is available in the China, and people eat that thing so that it got infected. I'm not going into details because much more important part will be there. But this slide is very important. Now, once it's got infected in the human, it takes some time. It can go more infection, and as being an RNA virus, its mutation rate is 600 times more than DNA virus. So it can mutate But if it mutated, where it is getting mutated, that's the most important part. And that's why the infectivity is high, how it is, I'm going to describe in the next slide. So, but just to conclude this one, there's a CoV1 and CoV2. Now, CoV1, you can see here, spillover reservoir is known, civet cat. You can call to break the chain. But in CoV2, still now, patient 0 is not known. Those who identify, that could be identified patient, but what it starts, nobody knew. So this is a debatable. So spillover reservoir is unknown. Second thing, for most transmission occurs in the hospital settings. When if it is in hospital settings and all those, so implemented barrier nursing is possible. But here it is now in wave 2, it's a widespread community transmission. So we don't know how to do for all those. And CoV1, the fatality is very high. No transmission was 24 hours to 36. So contact tracing effective. But here, most of the cases asymptomatic compared to wave 1 to wave 2, and some mild cases. After that is enhancified. So it's very difficult. So now, what is this? It's happening. Exactly what are the questions Dr. Shairam asked me. All of a sudden, if you see the wave 1 and wave 2, demand of oxygen has been enhanced. It is that patient numbers is from 1 lakh to 4 lakhs in the peak, number matters. But for individual cases, I think it has a consultation with a lot of doctors from where I have learned, and I'll be learning more. Dr. Swami is here today. From here, what I mean, things is even individual need of oxygen, the frequency, everything has been enhanced. Oxygen is not a medicine. Oxygen helps you to fight the disease for the lung and all. And so I'll go into the details for all those questions. But as of today, because of double mutant, all those things have been enhancing, viral load is more and all. Now, let me go a little slow, as others, all our chemists, all biologists told to go slow little bit for this virus of the mechanism. That's why I'm going little here and just want to tell you. This is the electronic microscope picture of COVID-19, what I got from Pasteur Institute of Paris. As I told, this is an RNA virus, and you know, there is a oily kind of a things are there, lipid layer, and these are the spike protein. And inside the plasmid RNA is there. Now, always doctor tells to destroy the virus, you wash your hand with the sanitizer, a 50% alcohol and soap. What sanitizer does? Sanitizer, if you wash your hands properly for 20 seconds, it will dissolve this lipid so that its genetic content, plasmid RNA will come out and the virus will be destroyed. But the question is, when you have a soap into 2 hands, how will you count the time? I'll tell you a little story, but I know these are legendary, all experts are here, but just to put it a little way, I'll tell you some story. Please bear with me. If you can count 1, 2, 3, 4 out of 24, then it is 20 seconds with a little gap. Then it will destroy the virus and you are okay. If it is not infected you, there is no questions of you are getting the disease. If you don't eat much, you will not, and if you don't expenditure more, you will not get body weight. So you don't have a baggage. So this is the doctors and all experts are telling, be very careful about taking care how to wash your hand and protect yourself from the mask and all those things. That is the first thing, what is the need of the hour for the students and everybody to follow strictly all those kind of things. Now, I have just here draw a picture just from this Pasteur Institute of Paris, what I got. Here you see, this is a diagram, pictorial diagram for this plasmid RNA genome. It has a genome here, plasmid RNA. I'll just come little later part, and you saw lots of lipid droplets are here, and these are all the spike protein. You see this spike protein like a scratch. If somebody some got heart or something need a scratch. It's like a tree, all those branches. And here, this is not through. It is just like a kind little bit of things are there. And I'm just going into for all those things. Now the question is, this is all those things are there and all. Now question is, how the virus gets inside the body. I'll tell you a story to you. You want to open your house and the house is locked. What you do? You have a key in your hand. You go there. You just take the lock in your hand and put the key in the group of this particular lock, but it will not open. Just putting into this, it will not open. What you do, you just hold it a little tightly with key and the lock. You just pull towards chest a little bit. Then you give a little bit of trigger, some energy, either right side on or left side. That's called the trigger. Then the key will be unlocked or locked, depending on which direction you are giving. Then you will get the entry. Take this example for this, exactly this virus, what it does. When it goes through the inside, through the nose and mouth and stays there for a little bit. That means after that, here I show a blue color. Let's see, this is this of a lung, some structure kind of a thing. And here there is a protein. Protein is there. Let's see, this is like an antenna. An antenna, when you have a TV, what you do? Antenna captures all the signals what is coming. Then it is got transmitted, you can see the TV. So what happens? This virus goes with this lipid layer, all those with just attach with the membrane. But that doesn't mean it has entered inside. It just attach into that. So after that, what happens? This, let's see, this black color, this is the lock, and this is the key. What are the example I've given? But what here in that case, when they come in contact with each other, you need some trigger to unlock this lock. Here in the body, you have an enzyme called furin. That particular furin cuts this particular there and make a continuous membrane, continuous tunnel kind of a thing. And through this, only this virus genomic, a plasmid mRNA will go inside your things, no, nothing else. And it can't replicate of its own. Being a plasmid RNA, it makes up its own protein and also makes its own copy, taking all ingredients from the body. That's why 1 to 2, 2 to 4, 4 to 8, like that, it replicates. Now the question is, how quickly it is going inside, and whatever the lock and key things are telling you, these are all amino acid sequence and protein-protein interaction or to bind each other. You can see here, this is just simply to make this thing to tell you, this is the amino acids, 1, 2, 3, 4, 5. Let's see, these are the parts of the key with a other way, which has to go inside the lock. So like that, it is happening 1, 2, 3, 4, 5, 6, that is called RBD domain. That is receptor binding domain with the SC2 protein, which has in that lung, it will go inside. Now, if this is the perfect sequence, it can go very quickly. But if it is not very perfect, it will take time to infect. Exactly in SARS CoV-2, what happens compared to CoV-1, the sequence is matching with this particular SC2 protein to interact very, very quickly. Now, after going inside, it became mutated, it became much more effective, much more powerful. That's why infectivity rate has been enhanced in the COVID-2, show us then after that replication and all those things happens. So in short, that's the way from mouth and your nose entering inside, it goes to the inside this lung and other organ, and it is creating a problem for the replication of that. But after entering inside oneself, after making its own copy or replications, it has to come out and go to infect in another cells. Like that it can go on, and more quickly, more efficiently it can do it. That is the disease pattern is coming. In wave 1, what happens, because of not that much of mutant, this effectiveness of the key to enter was a little different. Now it is more. We don't know in case of triple mutant, what's going to happen after that. So those are the things which are coming into the different paper and the expert biology. This is the mechanism what they're talking about. Now let's see here, this you can see in the left side here, after doing, it can go enter here inside, then it make its own proteins and all those, because it has to be replication and then translation again like that, and it has to infect for more of the things. And here there are a lot of questions for antibody getting raised to here, so that it should not infect or get the entry, or some other drugs, repurposing drugs are, I'll come into that. It is telling it is not very specific for that, but just to stop in the replication or it protein formation or agencies, so many things are there. So what World Health Organization, when the first wave came, they told that you took this repurposing of drug, but that is a very simple, those were attached to the clinical trial, and Swaminathan sir is there here, he will advise us more. The repurposing criteria strategy was very easy step. Strategy, non-blinded trial. Patients should not know whether they have received drugs or don't. And primary endpoint what they have, day the patient left the hospital or died, and duration of the hospital stay, whether the patient required oxygen or ventilation. This was the case when the first wave came. Now all doctors know, they have studied this virus, they know all those things. There are several repurposing drugs coming into there, tried to check the secondary infection and all those things and timetable it is making, so that it can't happen all those kind of a thing. But the problem is, what I just want to tell you a little bit, because of double mutant, and this time the viral load entries more, the duration of the replication very quickly is getting replicated. Its viral load is more. It is attacking your hemoglobin. Hemoglobin has no nucleus, but it is very surprising, it is attacking hemoglobin. And hemoglobin has two parts. One is the globin protein, another is the heme. Heme is made of iron-made porphyrin thing, whereas the ferrous is there. This COVID protein, after infection, it is just taking the place where the iron is binding to the hemoglobin, taking the oxygen and supply the oxygen to different parts of the body and organ, so that we are healthy. But as soon as the oxygen getting detached from the iron, and that particular place is taken care by the COVID protein, now the iron is coming into the free circulation. And as you know, free iron is very toxic. Body does two things. First of all, to capture the iron, what you do, ferritin level goes up, and all DJ dimer level disproportionately is coming. And then there will be alveolar specific, macrophage specific one cell, which does capture this free iron. It captures because it has to be in bound form, otherwise it will be a problem. But when it captures, at the same time, what it is doing this cytokine storm. Storm destroy everything. What is cytokine storm? Given just an example, let's say we all stayed in a colony, 150 houses are there with a security and gated community. Then in the night, street light is only on, others people, all they are sleeping. Then dacoits came, and they came and they want to loot all those things. First they have what they do, they make senseless the security people, but the entire all those areas very dark, there is no light. So in that case, when they go there, all of a sudden, after some time, the sense is coming back.
Corona gurinchi cheptundu vallu.
Parimal, you can unmute once again. Parimal, can you unmute? Parimal, can you unmute Parimal? Unmute. Yeah, you disconnected the presentation. Unmute. Now you can start again, please. It was some disturbance here.
Which slide?
Yes, 7.
Can we ask everybody else to mute?
No, I asked. I'm doing. We are not in the mute. I'm doing.
Which slide I should start, Venu?
7. Slide number 7.
Okay, fine.
No. I'm sorry. It is a slide number 10. Go
Slide
to the
number 10.
Go to the 10 directly.
Okay. All right. Okay. Is it okay?
Yes. Yes.
Okay. Fine. So what I'm telling, that sense of this security fellow has come back and he started shouting that all doctors has come to loot your house. You people come with all of your sword or whatever the things you have. So all those people, those things, they come out.
ఇది రెడ్డి ల్యాబ్స్ వాళ్ళు ఒక సైంటిస్ట్ ఇది జూమ్ లో మీటింగ్ పెట్టాడు ఇది కరోనా గురించి చూస్తానంటే నేను పాస్వర్డ్ అది ఇస్తాను.
You being the host, can you mute him?
Yeah, I'm trying. It's unable to do right.
Can I start, Venu, now?
Please go ahead. I'm sorry. Somebody.
No issue. So then, all of a sudden, this security fellow started shouting, all you wake up, come, that is people came to loot you. So everybody came out, but it is a dark. So everybody is attacking everybody. What happens, though some doctors, they got injured and left, but people killed each other. Same thing is happening for the cytokine storm. When the cytokine storm, for some cytokine level is okay, it does not that much. But all of a sudden, after the infection, when the hemoglobin this iron coming, it is captured by the alveolar macrophages. And when the cytokine storm starts, it attacking the lung. And you can see that after few days, some of this, you can see this particular thickening of the lung, the base part or some other parts, glass, so many things are happening. 50, 60, 70, 80% lung of the people are going up. LDH level is going, which is the marker for prognosis marker for different organ failure and all those, and people are dying very quickly. In another thing, what body is doing, it has to supply the oxygen to the organs. How to do that? To compensatory mechanism, it is enhancing this hemoglobin level, and hemoglobin level is going up. So you have hemoglobin going up, you have ferritin level going up, your D-dimer level is disproportionately getting regulated, you have LDAs, other things, and you are just about to have a cytokine storm. In that case, I just make a simple figure how it is happening to the irreversibly or fibrosis kind of a things in the lung, and people are dying very quickly. Here, doctor is telling we need frequent oxygen. If it is less than 92 or all those, immediately you give oxygen, because you need oxygen to keep your lung, everything active. So oxygen is not a medicine. It is just helping to keep you active. But here, lot of steroids people are giving. Those steroids enhance some sugar level and all those, but still they are giving lot of steroids to come out from this kind of things and all. So there is a lot of things of doctors are telling how to come out, but we need oxygen. If it is a crisis of oxygen, it could not going to survive this patient. And that's why most of the cases it's happening. One way, the frequency of oxygen is more. so the oxygen consumption more. And moreover, because of double mutant, this infectivity rate is so high, more and more patient. Adding together, need of oxygen has been enhanced, and this has to be given very, that will be our demand that each and every should get these things and all. Now, the things what I just told you, I'm going one by one. The key pathogen is step of substrate to attack the beta chain of hemoglobin, hunting the porphyrin, dissociating the iron from it, releasing the iron into the circulation. Thus, hemoglobin loses its capacity to bind with oxygen, so oxygen is not supplied to the major organs. That is why we see resistant hypoxia coupled with very rapid multi-organ failure. Moreover, the free iron released into circulation is so toxic as it causes a powerful oxidative damage. Free iron toxicity causes inflammation of alveolar macrophage, which leads to CT scan characteristics, HRCT, all those things we know. And the body tries to compensate this by elevating the rate of hemoglobin synthesis, which explain why hemoglobin rate is so high. Now, here there is a question before wave 1. There is a doctor prescribed, somebody doctor did not allow or convince. There is a debate the use of chloroquine. But you know, chloroquine, it protects the hemoglobin from the malarial attack. And the chloroquine, where it binds, this COVID protein binds there. So there could be a things to protect the hemoglobin after the infection. We don't know, Dr. Swami or doctors are there, whether it will be prophylactically or after this and when it will be done. But definitely chloroquine can protect the hemoglobin, and it can enhance the capacity, not so much of damage by this COVID, all those kind of a thing. So there is a thing here, the chloroquine phosphate competes with porphyrin, so chloroquine could be given for the infected people for certain dose. Second thing is coming for favipiravir. Favipiravir, you know all the mechanism of favipiravir, how it works and all. And this most ligand is 6-chloro-3-oxo, all those things what I have written there. The favi, though it is very weak, but it can stop the replication of the virus and all, because it binds to this particular 2007 times more compared to this porphyrin and all those things. So it will not allow the COVID virus to bind to that protein and damage it. So this is a kind of a things all experts people are thinking how to do for all those kind and come out from these things and all. So now here I just want to tell you, but there is another point it's coming, though it's a statistics. If you see the Dharavi case, 13 lakhs of some dwellers, those are poor people, and they do a lot of physical work, hard work for their own livelihoods and all those. When the first wave started, the Maharashtra government started giving a lot of measures. But what happens, Dharavi has not been affected. Death rate is not that much high. If you go to the slum dwellers of whole India, the statistics is how correct it is, we don't know. 10 to 15% slum dwellers are getting affected only. Most of the people getting affected, those who are middle class, higher middle class, or those who are lack of exercise kind of it, though it is not a herd, all those data. It is just something started coming. That's why telling India like things in the slum area, amount of or numbers of affected people are not that high compared to middle class and higher middle class. Though other doctors, when I talk to them, it may have some kind of a things, maybe the percentage wise, but there is no hard and fast rule that it will not affect and do it the way if it goes. This is one way I want to tell you. But another thing what doctors or everybody is facing, people are at home. Because of whom their life pattern has been changed. They are staying at the home for a long time. There are so many problems happening. All exams are stopped, and kids are they are taking rest in the morning, night because of internet. They are doing a lot of other things and all. Because of anxiety goes towards this kind of a stress, I work on stress-induced diabetes, so a little bit I can tell you about that. And the stress goes to the other kind of so many depressed, suicide, so many things are happening. But when you are stressed, that time your brain secretes a lot of hormones, which is not very good for your body. That is the one of the case. So how to get rid of that? When you think, your brain is always designed to think in a very on a linear way. But when you want to think something, you disturb your brain. But if you disturb much more, it will be a lot of stress there. And let's see, you are boiling some water, all vapors will come. If you put some cover on this vapor on the pot, the vapor will fill down with a drop of a water. Like when you're thinking for some bad thinking guy trying to get some kind of a solution, what happens? The thought process, if I can go in any other direction. If I give an example like a tadpole in the pond, there will be head, there will be tail, but there is no direction. And when it is more vibrant, some kind of a stream will be there, it will be moved further. Like that also our thought process goes like this. If you can take a pen and try to make this thought process to write something on your notebook. So what you are doing, what you explore, after writing through the pen on the notebook, you are consolidating, you are concretizing that things. Next day again you read, from there again you explore. So explore, consolidate, explore, consolidate. Like that, you can reduce your stress level. Number two, those students who are staying in most of the students of the India, they don't have an independent room, but those who have, if they can make their room a kind of a school kind of a concept, putting a legendary scientist sketch created by his own, or taking some legendary, all those people or Nobel laureate or some other field and keep it and change after 10 days, what happens? Their creativity is getting from the stress level towards. And in Hyderabad, lots of school people asked me, I just suggested one thing, when they're having an online class, please put your own uniform, the student, so that they can think of, though they're taking online class, but still they are at school. So those are the indirect kind of things are coming. But the point is, question, if you can walk 6 minutes, and after 6 minutes walking, just a decent speed, if your oxygen saturation level comes down 5%, you need to consult doctor immediately. But if you do exercise, physical exercise, all those things of then your vitamin D3 level should be enhanced a little bit. It's not like a sunlight, but 90% Indians, vitamin D3 level is very less than 30, sometimes even 5. It is a problem in the Middle East, for everywhere. It's a global pandemic and all those. And the third point, what I just want to tell you, those who are taking insulin to control diabetes, other than pill with insulin, those who are controlling diabetes, if they got infected severely with COVID virus, fatality rate is 27% higher compared to those who are taking pill. Because the insulin, all are linked with this, some other things and all. This is cell metabolism, clinical trial data, what I want to tell you. And here, Dr. Swaminathan there is a doctor, but I just, for my own knowledge, I want to tell you, those diabetic people who takes twice or thrice a tablet every day, if they got infected, they are taking several drugs for the COVID, doxycycline, secondary infection, then different kind of a steroid and all those kind of things. You need to see that how the things are getting excreted from the body. If it is getting excreted body to the kidney, then you are in a danger because your creatinine level will be going up. That is the comorbidities could be attached to your things and all. So those are the points. And now coming from the remdesivir crisis, I think Dr. Swami's doctor knows much more better than this. But as from my biology point of view and understanding, remdesivir inhibits this particular replication for exonuclease for RNA things to do that. But remdesivir can't, only when putting it, it can add for a replication one nucleotide After fifth nucleotide, it can do it. And remdesivir is not the drug always to treat this. It is a Ebola one, but here it is, we are giving the remdesivir, but we have to follow the doctor when it has to be given. Better the steroid. So many steroids are there, either capsule or injectable with the doctor supervision. That needs to be taken for all those kind of things and all. Now, coming back for the airborne, it is not confirmed. I think all the scientists here and my sir teacher, my colleague are here, they read that paper. There are 10-point agenda given. It could be airborne, but it is not confirmed yet. And moreover, a lot of things are running in the WhatsApp. What we need to discuss with the expert or the doctor, and with their permission, we have to go for all those kind of things. And about the vaccine, I think everybody knows here, there are 2 different times, and another vaccines are also coming. Everybody might have taken a dose or so. So there is a fear complex for taking vaccine. What going to happen, what is not there. We need to be vaccinated. But as of today, the Lancet data is telling either for Covishield or Covaxin or other things has to come, Sputnik or Moderna or anything, how much, how long it will protect, it is not known. So maybe after 6 months, I'm not very sure, maybe after 3rd dose could be taken after 6 months or 8 months, or we are going to a situation like a flu virus vaccine. Every year it's getting mutated and we need to change this vaccine for us. So that is the future, what we're going to tell now. But the 2 things what I want to tell, if virus doesn't infect us, then there is nothing. So first precaution, first is wearing mask, washing the hands with the sanitizer. There is a misconception for the common people. If alcohol can destroy the virus, if I drink alcohol, what is going to happen? Here the virus is on your palm, on your head, or everywhere, or on your hands and there, but they are inside, already inside these cells. So that to penetrate to that, to kill the virus, first this alcohol has to kill your cells, dissolve the lipid layer, then go to a second one. So it's not correct logic. Those are people doesn't understand, and that's why you see the long queue when that lockdown happens for all those kind of things. That's a different kind of things and all. So today's meeting for all those, my teachers, my friends, all are there from different parts of the world. Collectively, if we think and learn more, because every day hundreds of phone calls coming, death things are coming, want oxygen's coming, what to do. So maybe after discussing the question answer, getting the advice and the collective knowledge, all of us, we can protect ourselves better, or we can tell us and we can contribute a little bit to save the humankind. Thank you very much for giving me a chance to discuss with you all. And thank you, Shelva. And you started this thing. Thanks, Bhanu and Dr. Venkat, Dr. Swami, and all of my teachers. So now I will also take the question answer and I'll learn more so that I can understand better how to control this disease. Thank you.